After that, in the last embryo class, I told you all to prepare and come for presentation, right? Who all are prepared? Who all are prepared for presentation? I said you can use your PPT or you can use the board and you can discuss. Can you present?
So I will go discuss the spermatogenesis. Spermatogenesis is a process by which spermatozoa, that is the mature sperm, are produced from spermatogonial stem cells in the seminal tubules of the testes. So we all know that it starts at the time of puberty and it continues throughout the life. The sites are the seminal tubules of the testes. It is supported by the Sertoli cells. And the hormones that are that regulate it are FSH, LH, and testosterone. There are some stages of spermatogenesis. Mainly, there are 3 stages. The first one is spermatocyte, like the mitotic phase. The second one is meiosis. We have like 2 types of meiosis: meiosis 1 and meiosis 2. And then we have the last stage, that is Spermatogenesis. I'm gonna tell about the structure of sperm. The sperm has like 4 main parts that are head, neck, middle piece, and tail. The head is a very important part because there's an enzyme present that is echrosome that helps in the
What the function of echrosome? Like it helps in the head is going to insert in.
Yeah, during the fertilization, it is the first part that enters the.
During fertilization is the first part that enters the ovum. Then we have some hormones that happens for spermatogenesis, and I've already told LH, FSH, testosterone. That's it. And some of the factors that affects the spermatogenesis are high temperature. We all know that if there is a slight increase in the temperature, the process of sperm formation cannot occur properly. There are some other factors also like smoking, alcohol, radiation, etc. That's it.
Oh. You discuss oogenesis, please.
Hello. So basically, what she discussed is spermatogenesis, and now I will be discussing oogenesis a bit. So oogenesis is totally different from spermatogenesis. If you see, because oogenesis starts from the very beginning embryonic stage, and after the embryonic stage, there are no more formation of oogonia because we know levels of oogonia are found during the embryonic stage, and after that, after the March period, there are no more oogonia that will be forming in the body. So after the formation of oogonia, there are various processes through which the ovum is released, and I can show some processes through which the oogonia is completely developed into the ovum.
For these sessions? No, not yet.
So basically, what I've written is that the oogonia are the actual primordial cells or the germinal cells. We can say that is true, and after that, the oogonia starts mitosis and differentiation and gains into a much larger shape, which is known as the primary oocyte. So the primary oocyte will also be 2N. Then it gets partially arrested at the prophase 1 of cell division, and just prior to ovulation, it completes its first meiotic division. Then I've written is that it will form then the secondary oocyte, that is also meiosis. So basically, there are two meiotic divisions, and we can say one mitotic division. Oh yeah. Then the secondary oocyte also gets arrested at the metaphase stage of cell division, and just prior to the fertilization, it completes its second meiotic division, and thus it gets formed the ovum. So basically, what I have said is the cell structure. Now, if I say about the cell membranes and all, we can say there are four stages. That is the primordial follicular stage, and after that, the primordial follicular stage will get converted into the primary follicular stage, then the secondary, and then the tertiary, then the Graafian follicle, and the release of the ovum. So in the ovum, we can see briefly there are few major structures inside the ovum. We can say theca externa, that will be the outer covering of the ovum, and below that will be the theca interna, which will be formed at the Graafian follicle level, or we can say the tertiary follicle, which will contain the antrum also, because in the antrum, which is also a very important part of the ovum, because it's a fluid cavity. So after the formation of ovum, we can see a mature secondary oocyte there, and there will be corona radiata. There will be pre-midline space. So that's all. Thank you.